Hello, everyone, and welcome to today's webinar, Imaging Long-Range Neuronal Connections in the Mouse Brain by Light Sheet Fluorescence Expansion Microscopy. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Dr. Martin K. Schwarz, Group Leader, Functional Neuroconnectomics Group, Institute of Experimental Epileptology and Cognition Research, IEECR, University of Bonn. Professor Ulrich Kubitscheck, Professor for Biophysical Chemistry, University of Bonn. And Mr. Juan Eduardo Rodríguez, PhD candidate, Institute of Physical and Theoretical Chemistry, University of Bonn. Dr. Schwarz, Professor Kubitscheck, Mr. Rodríguez, you may now begin your presentation.
Yeah, thank you very much for this kind introduction. And yeah, it's my pleasure to start today's educational lecture. So you realize we have three speakers. Let me just give you some background why we have three speakers. And so how we arranged the talk. So I will, in the first few minutes, the first few slides, talk a bit about the motivation, the biological question behind why we are interested in imaging long-range neuronal connections. And then Ulrich Kubitscheck will continue with the basics of the technique, the basics about light sheet and light sheet expansion microscopy. And at the end, it will get really interesting. Juan will then will show you results, and we'll talk about the achievements that we did so far using these techniques. Now let me see. Next slide. It worked. So what is our motivation? So we are interested, so my group is actually interested in a very form of learning that is habituation. It's a very simple form of learning, and specifically to odors. I will come to this in a second. But what is also motivating us is a quote from a Nobel laureate, Sidney Brenner, who won the Nobel Prize in Physiology and Medicine in 2002 for C. elegans for apoptosis. But what he said, I found really inspiring, and we found, the group that is presenting today found really inspiring. It says that progress in science depends on new techniques, new discoveries, new ideas, and probably in that order. So he puts the progress, the development of new techniques at the forefront, and we would like to keep up with this spirit, and this spirit actually motivated the research and the topic that we're presenting today. So what is the topic in specific? We are trying to understand the connectomic organization of the wiring diagram between HDB, that is the horizontal diagonal band, and the OB, that is the OB, the olfactory bulb, that controls this simple form of learning that controls habituation. Now, right. Let me see. Next. Okay. So our interest is habituation, as I said before, and specifically to odors, but let me just give you a general idea what habituation is for those who are not that much in the field. So habituation is kind of it's a type of non-associative learning where a non-reinforced response to a stimulus decreases after repeated presentation of that stimulus. So what does this mean? Let me just give you a short example. So probably most of you, I have kids, and if I present my kids at home a new toy, so as a function of time, the more often I present the toy to this kid, the toy to the kid, it gets less interested. So it spends less time with this toy because he knows it already. So it means that the kid habituates in a way to this toy, right? So what is this good for? What is the consequence of this habituation? What does the community believe why this is important? We think that it's generally assumed that lowering a response to a stimulus that has no consequences frees up cognitive researchers to stimulate that might be more important, biologically more important. Now, how do we study the habituation in mice? And how do we study habituation to odors? So what we in principle do is we put the mouse into an open field arena, and in the middle we place, you see this on the right-hand side, we place a petri dish with an odor. And then we present this mouse to the 3 times to the same odor, right? And what you can see here then on the plot on the very right side, on the right-hand side, is that the latency increases as a function of the number of presentations, meaning that the mouse, the latency is the time the mouse takes to run from the corner to the odor, and this increases, meaning that the more often you present the mouse to the same odor, as a function of time, the mouse gets less interested. So it habituates to this odor. And to show that this is really habituation, just not fatigue, we present in the fourth run a novel odor, and you see in the novel odor the mouse runs very fast again in the middle of the arena because the mouse gets again interested. Yeah. So let me see in the next slide. Let me show you in the next slide how we try to figure out the circuitry that controls this type of learning, this habituation. And just to remind you, the olfactory bulb represents the first processing center in olfaction. So incoming odors are first processed in the olfactory bulb. So we thought it's very likely that the neurons that connect to the olfactory bulb, the neurons that influence the activity of the olfactory bulb, might be involved into habituation process that I showed you before. Yeah? And we check this by retrograde monotransynaptic tracing using rabies viruses. So what you in principle do is you inject this virus in the olfactory bulb, and what you see then is all the neurons that project to this region, so regulating this region. And on the right-hand side, you see already a picture that has been recorded with light sheet microscopy. That days it was called ultramicroscopy with conventional tissue clearing techniques. And what you see here, that there's very, very prominent projections from a region called HDB, horizontal diagonal band. In human, it would be the horizontal diagonal band of Broca that connects to the olfactory bulb. So indeed, we and others identified these type of neurons that regulate activity in the bulb. And in order to show that these neurons are also bringing about this phenotype, so the activity of these neurons, what we did is this is usually what you do, you can silence these neurons. You can delete this region, or you can silence these neurons by several different techniques. We use tetanus toxin light chain, but in principle, you take out, you inhibit the activity of these neurons that project to the olfactory bulb. And what you see here on the right-hand side in very blue is that odor habituation is completely abolished. So it means that this mouse does not habituate to the odor. It is like an Alzheimer patient, right? If you look into the mirror, and whenever you look in the mirror, you see a new face. So the mouse does not habituate, showing indeed that this region is causally linked or is controlling odor habituation in the mouse. Now, as a next step, what we did is we did a physiological analysis of the projection of these neurons, of the projection of these neurons into the olfactory bulb, and we did this by 2-photon calcium imaging of projections from the HDB within the olfactory bulb. And what we can, just to make a long story short, what we can show here is that indeed these neurons in the HDB are activated by odors, so they show response to odors, and moreover, they not only show response to odors, but they even show specific responses to odor. And this leaves us brings me already to the end of my presentation and leaves us with the question. So I could show you that the HDB projections are overtuned. I could show you that we could show that loss of HDB function abolishes olfactory habituation, and we know that the HDB is synaptically somehow connected to the OB. And now, what is still missing is the hardware that controls that controls this circuitry, and so we are teamed up with the team of Ulrich Kubitscheck to make a reconstruction of this region to see exactly who is connecting with whom, with the ultimate aim to generate computational models, realistic models to better understand their biology. And you see here, this is a formidable problem with very, very high resolution, but this will be explained by Ulrich in the next 10 minutes. Thank you very much.
Yeah, so what are the tasks and challenges that we face when we want to do such studies? As Martin mentioned, the most important point is that we need to image extended three-dimensional regions in the mouse brain. And secondly, we don't want only to image extended regions, but at a very high resolution, at synaptic resolution, in order to make out the details of the connections. And the problem is, I show you here at the lower left graph an image of the section of such a mouse brain. The problem is that it's opaque and that it's very large for microscopy. And so we have here a special technique that we need to use. And as the title already indicated of our talk, this is light sheet microscopy. So in order to remind you what the speciality of light sheet microscopy is, I made these two sketches here. You see in normal microscopy, the illumination occurs via the objective lens on the left, shown on the left-hand side. And the problem is that the complete extended sample is then illuminated and we produce a lot of fluorescence background. That can be overcome when we take the trick, or when we use the trick of illuminating the sample from the side, as is done in light sheet scanning microscopy. And we see that then we illuminate only that region that is finally being imaged. So we illuminate only the focal region. The idea of this technique is pretty old. It was invented by Zsigmondy and Siedentopf already in 1903. It was revived in the 90s of the last century, and then really put to a new use by the work group of Ernst Stelzer together with Jan Huisken in 2004. And since then, the number of publications using this technique is really exploding. In order to demonstrate what the advantage is, we have these two movies. At the left-hand side, we see simply a solution of fluorescent molecules in epifluorescence illumination, and you see that large background, and we can see, using epi-illumination, normal fluorescence microscopy, single molecules that diffuse around in the solution, but the contrast is bad and the background is high. When we use sheet illumination, as seen now on the right-hand side, we see that the contrast incredibly increases and that we really can make out without any problem single molecules that diffuse around in that solution. So sheet illumination really opens the way to produce optical sections in extended samples that need to be imaged fastly. If we compare this technique If we compare this technique with confocal laser scanning microscopy, for example, as shown on the left-hand side, We see that confocal microscopy provides low frame rates only. It has a high photo damage because we illuminate for each image the complete sample. We produce a lot of out-of-focus fluorescence, but that is then finally removed by the pinhole that is normally used in confocal microscopy. On the other hand, light sheet fluorescence microscopy can be used with a very high frame rate because we can use a parallel data acquisition using microscopic high-speed cameras. The imaging method has a very low photo damage because we only always image those fields and we illuminate only those fields that we indeed image. We perform no out-of-focus excitation, but the technique is sensitive to light absorption and to scattering light. So the latter problem can be overcome by using clearing techniques. For example, as it is provided by expansion microscopy. I come to that in a second. So here we see in the upper sketch excitation light path of our microscope. Actually here we see on the right-hand side a very complex light path which is used for airy beam or lattice light sheet illumination. We do not use that part in the experiments that I will show. We only use a normal Gaussian illumination that is then scanned along the focal plane in order to create in the time average a light sheet. The sample region illuminated by that light sheet, as shown below, is then imaged by an objective lens. The focus of that lens is in the region where we illuminate the sample, and that image is then mapped onto a 10 megapixel CMOS scientific camera. We have the option to use several objective lenses. We have a dipping system, and we have one home-built system, which is used for very high resolution experiments and for experiments with a lower resolution where we need a higher data throughput. There we use a Blaze from Miltenyi Biotech. So far we saw how we can acquire with high speed optical sections from samples, also from extended samples, because with that fast imaging technique we can achieve high frame rates. But still we didn't talk about the problem how to reach the necessary resolution, because in such samples we have to use usually low NA objective lenses, and these have a low optical resolution. So what options exist in order to image points of interest that are closely together so that their diffracted images overlap, as shown here in that sketch? One well-known approach to solve that problem is to use a point spread function which is simply sharper than the normal one. This is provided by techniques like stimulated emission depletion or SIM or STORM. So that is one option, but the problem with these type of techniques is that they don't reach deep into the sample. Due to phase problems, these techniques only extend about 100 micrometer at maximum into a extended sample. So what you also can do to solve that problem is not to sharpen the PSF, but simply to expand the sample. In that way, you guarantee that the distance between the points of interest becomes large enough in order to be resolved. That technique was invented some years ago and was introduced by the lab of Ed Boyden. Here I show you in some simple steps how that expansion techniques in order to magnify your sample works. We see here a single cell with some regions of interest. These orange dots in that image, you see a very high magnification of that on the upper part of the image. And the steps that we need in order to produce an expansion of that system is simply first by labeling it with fluorescence, in this case, for example, by a two-step immunofluorescence labeling. So we achieve a distribution of the fluorescent labels on our epitopes. You see that in the magnification on the right-hand side. The next step is then to link the many chemical residues in these proteins with bifunctional linkers that then in the next step can be connected to a gel. So after that linking process, we introduce a polyacrylamide gel actually in our sample, and the linker connects to the polymers of this gel, and on the other side to the proteins. In the next step, we go along and digest the major part of the proteins, not completely, but the major part of the proteins, so that actually the overall structure of the sample is lost, and we are left with the fluorescent labels linked to that gel structure. So we don't have the proteins anymore. We have some peptide residues labeled with the fluorescent dyes, and these are linked by the bifunctional linkers to the gel. Already in this step, the sample becomes transparent. Usually it expands already by a factor of about 1.5. But if you now exchange the digestion buffer, PBS is the usual buffer used in this situation, by distilled water, you find that the sample becomes largely expanded, usually by a factor of 4. So that is, in some brief words, the technique of expansion microscopy as introduced by Ed Boyden. So this approach has two key advantages. First, we obtain a transparent samples with a refractive index of water. And secondly, actually, we can also do post-labelings in this situation and can, in that digested sample, try to achieve additional antibody labeling. And the digested sample provides a better penetration of such antibodies. So I show you now an example of this, where we applied this expansion technique to organoids, brain organoids. And you can see on the left-hand side, the upper image, the organoid, 2 months old, and you see the tiny white dot in the center that is opaque. Then in the center image, it is already digested and transparent, and then it expands on the right-hand side in the, you see the magnified gel. And then finally, we can image that with a light sheet microscope. So here I show you one example, what can be achieved using this technique in organoids. So we have a sample that is in size several millimeters large, and then we can zoom into that sample using light sheet microscopy, and we can zoom in so well that we finally achieve synaptic resolution, as shown in the lower images that you finally can also deconvolve. Okay, this technique now we applied to thick mouse brain sections, and that will be described by Juan, and also the results that we achieved in this manner.
So thanks. Well, here in this slide, you can see in the upper part, like this sagittal cut of the mouse brain that is 2 millimeters thick, and as Ulrich already explained, is opaque, so it will be really difficult to image through the sample. So what we did was we optimized the technique in order to have like a completely transparent sample, and then overcoming the problem of the scattering if we want to use light sheet. That is like probably the most adequate technique to image this kind of samples. In the lower right part, you can see like a sagittal view corresponding to exactly the same sample, where we have in blue nuclear staining and in green GFP. And there is a reason why we are also using sagittal cuts, because there is a more natural way to follow the projections of these neurons from the HDV to the OB, compared to what is more traditional to use coronal sections. So now that we know that the sample is working properly in this kind of samples, so then we go back to the original problem, that is labeling different groups of neurons in the HDV. So the HDV is mostly made by inhibitory GABAergic neurons, modulatory cholinergic neurons, and excitatory glutamatergic neurons. So what we did, and this was done by Irina Pavlova from Martin's lab. So we took 3 different mouse groups that were injected with a virus that will express GFP in the complete neuronal cell body, and in the presynaptic part where is synaptophysin accumulated, we will be expressing mRuby. So, all right. Let's see if the controls. Okay, so now you can see the sagittal view, right, of one of these mouse brain samples. And this is a maximum intensity projection comprising 200 images. And this was acquired with the Blaze microscope with a 4x magnification objective. So you can see in the details of the image that this image is already about 15 millimeters long and 5 millimeters in y direction. If we have the complete volume, then we are able to recognize some important parts of this circuit, as I'm showing you here in this part. So we can clearly see the injected neurons in the HDV. We can also follow the fiber path that is going from the HDV to the olfactory valve. And even if in this image is not that clear, if we zoom in and we focus in the olfactory valve, then we are able to see all the fibers that are connecting there. So if you see the scale of the image, you can see that this circuit is already here about 6 millimeters long from the HDV to the very end of the olfactory valve. So that is a difficult task to achieve with a different kind of technique. But now if we want to focus and see where the neurons of the HDV are projecting into the olfactory valve, right? So we will focus in the olfactory valve. We have the complete olfactory valve that was imaged with this Blaze microscope and the 4X. But as you saw in the previous image, it's not that easy to recognize or to analyze the fibers, because they are too small and we don't have enough resolution. So as Ulrich explained, we can change objective in the microscope, and using exactly the same sample, then we can go with a high magnification objective and image a volume that contains the olfactory valve, as you can see there in yellow. And if we take like a sagittal perspective, we are able to see all the fibers that are going into the olfactory valve, but also with the same volume and with the same sample, we can also get the we can make a coronal view. And that is really useful for us because, as you can see there, we can recognize the internal extractor of the olfactory valve, and we will use that to analyze where the fibers are projecting. So having that in mind, so in the coronal view, you can see that if we make a line crossing that, we can recognize at least 3 of the main parts of the olfactory valve, that is the glomerular layer, the external plexiform layer, and the granule cell layer in the middle. And then we have again external plexiform and granule cell layer. So if now we take in this volume like a projection across that volume, and we measure the mean intensity value. For example, with the nuclear staining, we have that graph. There we can clearly identify these three different regions, the glomerular layer, external plexiform, and the granular cell layer. And we can apply that now to the intensity, but in the GFP signal or in the GFP channel. So we can see that most of the projections in this sample, that is a GABAergic sample, so most of the projections are distributed like to the center, like closer to the granular cell layer, and little in the glomerular, and even less in the external plexiform. So with this, we analyze three different mouse of each group, so 3 mouse of GABAergic, cholinergic, and glutamatergic, and we got different distributions. So for the GABAergic, we can, as I already showed you, we can see that most of the projections are concentrated in the granular cell layer in the outer part, so probably the mitral cell layer or something. So we have like 60% more projections in that area. But if we go to the cholinergic, we can see that the projections are more concentrated in the external part, that is the glomerular layer. And finally, for the glutamatergic, we can see that projections are mainly, like more than 80% of the projections are just going to the middle part or to the central part of the granular cell layer. So that means that maybe we can this give us a hint that like different neural types project to different regions in the olfactory valve. That was what Martin was mentioning in the beginning. And so then we have like a different recruitment of the network inhibitions there. And that's something that only we can see if we look to the synapses. And that is the next step. So here we have, again, a sagittal view in the olfactory valve that was acquired with the place. And if we zoom in, we can see little dots in magenta in that magnified region. But as you can see, one thing is good is that we can preserve this small amount of fluorescent proteins that was in the synaptophysin part, but we don't have enough resolution to identify this presynaptic marker in 3D. Okay, that is important. So then we change exactly the same sample. We put it in our custom built light sheet where we can fit like a higher magnification objective, and there you can see that it's easier or more clear to identify these presynaptic markers. So to be sure, we can even go further, and as I'm showing you here, like a magnification of that region, but now with 100x objective, and we can see that it's actually not an artifact that we have like that colocalization in the previous image, but that actually the presynaptic mark is precisely located in these synaptic buttons in the projection. So now, if we take a volume, right, with this high magnification objective in the olfactory valve, and I'm showing you here the example of the GABAergic, of the glutamatergic mouse brain, where we have more like the connections in the center of the granular cell layer. If we take a volume there with this high magnification objective, we should be able to see that the number of synapses, so if we count the synapses along that volume, we should be able to see that while we are going deep into the sample, the number of synapses should increase. And that is what we quantify and what you can see there in the right part of the slide. So we are able to identify precisely where the synapses are, like taking into account the colocalization of the marker, this mRuby or this synaptophysin marker with the GFP signal. And we did that, of course, for all the images and for all the mouse groups. And we found that there is a main distribution in the granule cell layer. The cholinergic ones also project a lot and to the glomerular layer, but we can see, for example, the glutamatergic have almost no connections in that area. So finally, we can say that the distribution of different types of neuronal fibers from the HTV to the OB suggests a differential and neurotype-specific recruitment of the network inhibition within the framework of the olfactory habituation. Now, as a summary, or more technical summary of the technique, so we can use this technique to go from the centimeter scale to the nanometer scale using one sample. So you don't lose information at all. We optimize this kind of protocol for like really extended brain slices. We can preserve, even after like really extended digestion cycles, all the autofluorescent proteins. And with this, you are able to do the analysis of neuronal projections from the HTV into the olfactory bulb, as I already mentioned, also looking into the distribution or spatial distribution of the synapses. So that's all from our part. I would like to acknowledge all the people from the biophysical chemistry group, and of course from the group of the experimental epileptology and cognition research. So thanks for listening. So we are open to questions now.
Thank you, Dr. Schwarz, Professor Kubitscheck, Mr. Rodríguez, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, how long does the complete preparation of the mouse brain section take for the expansion process?
So a sample like the one that I show takes, the complete process is about a week. So between like sample preparation, and the part that takes longer is actually the digestion part that was mentioned in Ulrich, where we have to remove all the lipids and start to break all the proteins to make the sample transparent.
I see. Thank you. Next question. How long takes a typical data acquisition session, let's say from the complete volume of HDB and OB at 4x magnification?
That usually takes like about one day with the Blaze and the 4x, especially because we have two different channels. So and that's more or less the average.
All right, thank you. Next question. Thank you very much for the nice talk. So in expansion microscopy technique, there's no requirement of clearing solutions.
Yeah. Yes. No. So.
The heavy absorption of water.
Yeah.
The heavy absorption of water.
Oh yes. I mean so what makes a sample not transparent is basically the amount of, are the lipids. That's the main source of things that, and the proteins, and that's what we are removing with this digestion buffer. So finally, then when you want to image the sample, you can place it either in PBS to keep it to expand it 1.5 times, or put it in water. The advantage is that you don't need any special kind of objectives, so you can use any water immersion objective, and you don't need a special sample chamber for that. So there is no clearing solution there.
All right, thank you. Next question. How long does it take to image one OB sample with 2 different fluorophores?
The complete olfactory bulb section with, for example, with DAPI and GFP, with the 12X is more or less half a day.
All right. Next question. Oh, sorry. Did you have more
that,
to add?
That will depend always in the type of magnification and what do you want to see in the sample. So like you can also go like with a really high magnification, and then it will take longer because then you have to reduce the step size. You will have more tiles that you have to stitch and so on. So it's that's why I'm giving the example with that 12X, just to have to give a rough idea of that.
All right, thank you. Next question. How do you optimize the sharpen algorithm to improve resolution and to reduce the fake noise signal at the same time?
Could you make the question again, please?
Yes. How do you optimize the sharpen algorithm to improve resolution and to reduce the fake noise signal at the same time?
Well, maybe that's something that I forgot to mention. But all the images that I show now, they are raw images, so they don't have any post-processing procedure. So they are just, they are of course stitched because we take several tiles, but the images that I show are not after deconvolution yet. So the idea was to show what you are able to get just applying the technique and getting from the microscope out.
Great, thank you. Next question. Go ahead.
So maybe one should add that using the CMOS cameras, they are cameras with a very, very low noise. Yeah, and of course, what we always have then finally is a shot noise signal. But this is so low because our labeling is pretty extensive and pretty optimal. So we do not really have the problem of dealing with a lot of noise generally. In cases where we have really low labelings, this might happen. And of course, then you are limited in achieving an ultimate resolution because of the bad signal. But this is a general problem.
Great, thank you. Next question. Do you use anti-fading reagents in your PBS water when imaging? And if yes, which ones?
Anti-fading?
No. It's just PBS with sodium aside, usually to avoid any growing thing there, but just PBS.
All right, thank you. Next question. How heavy are the images obtained? How to handle heavy images?
Yes, that's true, and that's why it's also recommended, or that's why we jump from different objectives depending what we want to image. So yeah, I think that here Ulrich could explain.
Yeah, we expected such a question. So the amount of data we deal with in light sheet microscopy is always really a key issue. And here we prepared the table, and you can see that on the right-hand side, of course, depending on the resolution you are imaging with, you produce an amount of data which is pretty large even for nowadays computers. So with the ultimate resolution that we can obtain using a 25x objective lens with an NA of 1.1, then in order to image a sample of 1 millimeter to the power of 3, will produce at highest resolution then a data amount of 8 terabytes. And we stream the data from the microscope directly on a storage, on a hive that is directly standing beside the microscope. And we stitch this data then together because, of course, we have to image these large extended samples and with a lot of single tiles that are then fused together. We do that by using Fiji normally. The publication by, I think, Stefan Preibisch et al. So we use that device. And then we import the images finally into Imaris, and there we do the processing and the evaluation of the data. So all that is an own very special and quite a demanding task for the computers that you use.
Oh, wow. Thank you. Next question. Thank you for your informative presentation. How long can the autofluorescent proteins be preserved?
This is a very excellent question because it's of great practical importance. But Juan has a good answer to that.
Yeah. If you keep the sample on PBS, at least what we experience is we can easily keep a sample in good conditions for up to a year. So if you have to, of course, change PBS from time to time. But if you keep the sample in water, then it's a problem because fluorescence, like the protein, start to degrade over time. And then you can lose the fluorescence in 1 or 2 weeks. So what we do is usually if we need to expand the sample, we expand it for the imaging session. And after that, you can take the sample, you can shrink it again if you start to wash it with PBS. And then we keep it in PBS for longer incubation.
Thank you.
Frozen or not frozen?
No, at 4 degrees. Yeah, you cannot freeze the sample because you have a polymer gel that is filled with water. Then if you put that in minus 20, it will crystallize and everything is gone.
All right. Next question. How long is the preparation suitable for imaging? Does it fade out or degrade?
I think that's related with the previous one. So if you image with the sample in PBS, you don't have any problem. So I at least I have the experience to imaging the same sample like really long, just like for testing. But with water is then you will see the degradation over time.
Thank you. Next question. Would you tell us how the dendritic spines and their heads and necks are observed in your experiments?
How the dendritic spine?
Would you tell us how the dendritic spines and their heads and necks are observed in your experiments?
We didn't do any analysis on the spines, like the shape of the yeah. I mean, you can see the slides. So we did that for brain organoids, but for mouse brains, we didn't do that analysis. Was not the focus at least this time. But it's possible to do because you will have enough resolution and the 3D information to do that kind of analysis. So if you take a sample like the one that we are showing in the lower right part in Imaris, you can use this tracing tool that is available to count and then recognize the spine neck.
Great, thank you.
So here we have mainly the presynaptic marker. So that's what we wanted to show. So that's what we stress here in this graph.
All right. Next question. Which final optical resolution can be achieved in your microscope?
I mean, now if you expand 4 times the sample, like if you put the sample in water, here you can see what we have is an optical resolution of 300 nanometers. Like if you see the nanoscale, like the last row, with the 25 objective plus the 4 times expansion, we can achieve like a 300 nanometers in X and Y and axial 1 microns. That means that you have to divide that resolution by 4. So we have about 100 nanometers resolution in lateral resolution and 300 in axial resolution.
So we call that a virtual resolution, yeah, because I mean the expanded sample, of course, you have to resize, and that I mean you don't really improve the optical resolution. This is clear, but you get a virtual resolution in your sample because it is simply larger. And so we really come down to values that normally are considered to be super resolution. Yeah, these 0.1 micrometer. This is also achieved by structured illumination microscopy or area scan confocal microscopy. Not even actually, yeah, but we come close. So we are even better than these techniques.
And I mean, you can also use these iterative protocols, right? So not only expanded by a factor of 4, but expanded by a factor of 10. Which would then, or a factor of 20 right now, right? Which would then allow you to come down to a lateral resolution of 20 nanometers in principle.
Great, thank you. Next question. Given the odor-specific activation of HDB fibers within OB and the fact that the HDB does not receive direct input from OB, along which pathway is OB output relayed into HDB?
Yeah, this is a biological question. It's a good question. Actually, the HDB does not receive direct input from the OB, and the question is how is this odor information related to the HDB? And we did also monotransynaptic tracing and found that the OB projects into the olfactory amygdala, and the olfactory amygdala projects into the paraventricular nucleus, and from the paraventricular nucleus then goes the projection to the HDB, which is then relayed into the olfactory bulb.
Wow. All right, thank you. And it looks like we have time for one more question. Why do you not image just the complete sample with high resolution?
Yeah, I mean, as we mentioned, and as you can see in this table, going with a really high resolution to image the complete sample will be just like too much data that will not give you actually more information than the one that you can go get if you like kind of partialize your imaging session. So I think that the advantage of this kind of light system, the one that we show, like the place where you can change objective, plus the fact that you have like entire neuronal circuit contained in your sample, is the one that actually allows you to get all the necessary information, but in a, let's say, a smarter way.
All right. Well, thank you, Dr. Schwarz, Professor Kubitscheck, Mr. Rodríguez. Do you have any closing statements for our audience?
Yeah, maybe that since some years we are working on these techniques and developing our microscopes in order to optimize that. Martin is working on the labeling, and this is Juan is doing the experiments and the data evaluation. So this is really a wonderful example for interdisciplinary science, yeah. And we think we are so successful because we collaborate so well, and that works so nicely all together. So in case people are interested in applying and using these techniques, we are open for contacts, and they may just address us, and we might lead further discussions, and we are open to that, absolutely.
Nothing to add. This is keep up the spirit of Sidney Brenner, right?
Well, thank you again, Dr. Schwarz, Professor Kubitscheck, Mr. Rodríguez, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Till next time, take care, everyone, and goodbye.